wanted to go over that again. And everything, remember, this is a graph, so everything you need to really know is, well, not really everything, but to understand it, the information is there. That's what a graph does, is it's a pictorial delivery of information. So, remember, even though this ends at 100, we're not talking about percent. So on the x-axis here, that value is PO2. You're going to have a graph of this on your exam, all right? And so it'll be the two lines and the normal oxygen dissociation curve, but I'm not going to tell you that this is PO2, and I'm not going to tell you that this is hemoglobin saturation. I want you to be able to figure out which one is which. And just because they both end at 100, obviously, as I said yesterday, we can't get higher than 100% saturation. So this value tops out at 100. This value can go up to 120 or 130 if we're breathing in pure oxygen. So this is a measure. Well, you tell me, what is PO2? How do we measure arterial PO2, not alveolar, but arterial PO2? What is it a measurement of?
Oxygen in the plasma.
All right, so it's the gas molecules interspersed amongst the water molecules of the plasma. It's still a gas. It's still acting as a gas, which, you know, it moves around, it bumps against other water molecules, it bumps against the blood vessel walls, and it exerts a gas pressure. But again, remember I said if that's a difficult concept, change these, instead of a gas pressure, change it to mentally a concept of individual molecules. So as we increase from a PO2 of 40 to 80, we're increasing the number of gas molecules dissolved in the plasma. All right? So, again, when we talk about the shift to the right or the left, we choose an arbitrary point of 50% saturation, and we compare the two. So if we look at the fact that at 50% saturation with our normal oxygen dissociation curve, it takes this amount, and it's about typically 40, PO2 of 40, to have 50% saturation of hemoglobin. Those numbers you don't have to memorize. But as we shift our curve to the right, what are we doing in the value of PO2? Are we adding more oxygen molecules? Are we at a level now of more oxygen molecules in the blood or less?
More.
More. Okay? So we're in a situation here where we have our hemoglobin molecule, and it's going to have oxygen bound to it. And when we shift our curve to the right, we're saying that oxygen separates and comes off of the iron and moves into the plasma and then moves into the tissues more quickly. There's a physical change in the chemical interaction between iron and oxygen, so it does not stay attached to each other very long. So when that happens, we have less hemoglobin saturation. To keep the same amount of hemoglobin saturation, we need to add more oxygen into the plasma so that any time this comes off, another oxygen is right there to take its place. Otherwise, at any given snapshot moment, we would not have the same 50% saturation. So we're saying to keep the same 50% saturation, because how easily it comes off, we have to have additional oxygen in the plasma to take its place. So it's not that under higher temperature there is more oxygen. We're saying that because of the lower affinity, to keep the same saturation, we would have to have more oxygen. This is a good thing, all right? When you think about why we would have increased temperature, why we would have a lower pH, is because of increased cell activity, using up the oxygen. So the fact that the oxygen has a lower affinity and comes off more easily is better for the body. Okay? It makes it easier for the oxygen to get, for the tissues to get more oxygen. So then when we shift our curve to the left, we're saying we can get the same saturation with less oxygen dissolved in the plasma. That indicates the oxygen is staying on the hemoglobin ion longer. Okay, so we don't need as many oxygen molecules dissolved in the plasma to keep that saturated because the oxygen is not coming off. So colder temperatures, indications where we don't need the oxygen as much, or we have fetal hemoglobin. Why does carbon monoxide affect this?
It takes the spots of the oxygen.
Two things, all right? So it binds to the same place that oxygen does, but then it also prevents oxygen from leaving. Just remember, the last oxygen to bind to hemoglobin is going to attach to it more easily, therefore it's more difficult for it to come off. Well, that means that first oxygen is not going to come off as easily because carbon monoxide is there. So we're going to have it staying on longer, hence our curve shifts to the left. Okay? All right, that brings us, before we move on to carbon dioxide, that brings us to a discussion of hypoxia. And hypoxia is a condition of low oxygen in the tissues. You guys remember from Bio 430? We'll do it again our last lecture when we're talking about oxygen being used by the cells. We'll have 1 day left in class. All right, so for the entire semester. So where's oxygen utilized in the cell? Why do we need oxygen?
Mitochondria.
In the mitochondria. So when we talk about glycolysis and citric acid transport and a citric acid cycle, which is in the mitochondria, and then finally electron transport, oxygen comes in at the very last step to accept the electrons and hydrogen ions in the formation of water, after we've gotten as much energy as we can out of the hydrogens and electrons. So the cells need oxygen in ATP synthesis, right? So that's what hypoxia is that lack of oxygen to the tissues. So this individual here, you can see how dusky the fingertips are. That's the way my hand looks when I get cold. I have constriction of the arterioles in my hand. And that's an indication of lack of oxygen to the tissues. Cyanosis, cyan is a blue color. All right, and so that's showing low oxygen in the tissues. And in your lecture notes, 4 causes are listed of hypoxia. So as we look back at what we've talked about in oxygen delivery, remember we had pulmonary ventilation, we had external respiration, and we're finishing that up today with internal respiration. We kind of talked about both of those with oxygen. Today we're looking at carbon dioxide. So we can have problems. Any of these are either lack of oxygen available, a lack of hemoglobin to carry the oxygen, a problem with the blood vessels or the heart to move that hemoglobin, or a problem with the tissues utilizing it. Okay? So the first case is a problem with the hemoglobin. So there's nothing wrong with the amount of oxygen in the alveoli. There's nothing wrong with the diffusion. We don't have pneumonia. We don't have interference with diffusion of oxygen into the blood. What we have is a problem somehow in the transport, what's carrying that oxygen. So the most common is some form of anemia. So not enough red blood cells, or not enough iron, if we have enough red blood cells, or not enough protein to hold the iron. Okay? So in that case, we would have a normal PO2. Why would we have a normal PO2 with anemia? What is PO2 again?
Oxygen in the...
It's the oxygen dissolved in the plasma, and that's dependent on the oxygen moving from the alveoli to the blood. We don't have a problem with that, right? We don't have pneumonia. We don't have pulmonary edema. We don't have emphysema. So the oxygen is moving easily from the lungs to the blood. There's just not enough iron or hemoglobin for that oxygen to bind to. And that's where we need most of our, that's how we transport most of our oxygen. Remember I said we didn't have enough hemoglobin? The oxygen dissolved in the blood would not be enough to keep us alive. So we need to have that, a normal amount of hemoglobin. So a low hematocrit or a low hemoglobin, would that hemoglobin be saturated?
No.
Let's say that I normally have 15 grams of hemoglobin, okay? I probably have about 12, but ranges around 12 to 16. And we have normal PO2 in the blood, so we have adequate oxygen. All of the hemoglobin we have is going to be saturated. But if I only have 10 grams or 8 grams, that will be saturated,
But it's not enough.
but it's not enough for the amount of oxygen that the tissues are needing, okay? So I would have low hemoglobin, all of it would be saturated, I just wouldn't have enough of it. Okay? Carbon monoxide poisoning, we were just talking about that. All right. We would have a normal hematocrit because there's nothing wrong with the amount of red blood cells we have. Red blood cells are normal. Our PO2 is going to be normal. But our saturation is low because 25 or 50 or 75% of those iron ions are binding carbon monoxide instead of oxygen. So if we have 1 out of every 4 iron ions is binding carbon monoxide, we would have only 75% saturation. Okay, so we would be losing 25% of our oxygen.
What's the AV?
That's arterial, the difference between the arteries and the venous concentration. Okay? So we would have a very low, we would have a normal arterial PO2, but we'd have a very low venous PO2 because we'd have to be taking, normally it's 40 millimeters of mercury because that's being replaced by oxygen coming off the hemoglobin. As the tissues are using oxygen out of the plasma, oxygen's coming off the hemoglobin. But if oxygen's not coming off the hemoglobin, we're going to use more than, we go from 104 millimeters of mercury to 40 as far as the number. So if you change it to numbers arbitrarily, we have 100 molecules of oxygen. We use 60 of them by the time the blood gets through the tissues.
Okay.
Well, because we keep replacing that with oxygen coming off the hemoglobin. If the oxygen is not coming off the hemoglobin, we're going to use more of the oxygen that's dissolved in the plasma. We're going to be down around 20 or 15.
Okay.
Okay? So any other factor that shifts the oxygen? So let's say somebody has, we have a guy who through some genetic disorder has a high amount of fetal hemoglobin. He's still making fetal hemoglobin instead of adult hemoglobin. What's the difference between fetal hemoglobin and adult hemoglobin? Fetal hemoglobin shift our oxygen dissociation curve to the left or to the right?
To the left.
To the left, which means it's going to hold on to the oxygen longer, okay? So even though we have a normal amount of oxygen bound to the hemoglobin, when it gets to the tissues, it's not going to come off as rapidly. So when it returns back to the lungs, there's still going to be oxygen bound to that hemoglobin. All right, so that's the hypemic. Emia refers to blood vessels. We have a problem with the blood. Okay, this is just talking about carbon monoxide. Stagnant hypoxia. This is a problem either with blood vessels or with our pump, with the heart. So diffusion, the amount of oxygen in the air that we're breathing is okay. Nothing wrong with that. We still have 104 millimeters of mercury of oxygen in our alveoli. Diffusion distance is okay. We don't have pneumonia. We don't have pulmonary edema. Have a normal amount of red blood cells and a normal amount of hemoglobin. But we have a problem with maybe the heart is in congestive heart failure. All right, it's not moving the blood rapidly through the body in a rate to the use of the oxygen. So the body is using up the oxygen at a rate faster than the heart is pumping blood to that area with additional oxygen. Or we get blocking off a pulmonary embolism is going to decrease the amount of blood going through the lungs to pick up tissue, and there's some kind of problem with the delivery of that. Notice how much redder this individual's legs are, so that's that pooling. It's not getting back to the body to pick up oxygen. So there's decreased overall volume of blood that's moving through. So this is a vessel or pump disorder. Okay, this is what we talked about in Bio 430 in my class last semester, we had a case study of the dog getting potassium cyanide poisoning by eating some poisonous plants, and his owner tried to resuscitate him and got cyanide poisoning himself from the plants. So potassium cyanide inhibits the enzyme in the mitochondria that utilizes the oxygen. So the cells are hypoxic. None of the rest of the body is hypoxic. We're looking at, there's no problem with diffusion, there's no problem with transport, there's no problem with the amount of oxygen that's in the air that you're breathing. It's at a very cellular, the very last step where the cell needs to use the oxygen, it can't. All right, because of poisoning by alcohol or by potassium cyanide.
So the alcohol and potassium cyanide do the same thing?
Similar. Yeah. All right, so then the last slide is really what we tend to think of when we think of hypoxia. And that's not breathing enough oxygen. Okay? So that term is called hypoxia. So when I was doing my master's down at Loma Linda University, I was in the anatomy department working with fetal physiology. The experimental animals were actually guinea pigs. And a series of experiments were done. The first ones were the pregnant guinea pigs were exercised for 45 minutes a day. And then almost term, the moms were anesthetized with a lethal dose, but while they were still alive and under anesthesia, chemicals were used to measure the diffusion rate of oxygen across from the maternal blood to the fetal blood. How fast did the oxygen diffuse? And then while the heart was still beating, the placenta, chemicals like glutaraldehyde, a form of that, was added to the uterine artery and went into the placenta and caused it to be fixated at a normal blood pressure and volume. So the blood vessels were the same normal size that they would have been. They weren't constricted, they weren't dilated. And then we further fixed them, sectioned them, took pictures of them, and I used a computer stylus or pen to trace the blood vessels and see the number of fetal blood vessels and maternal blood vessels, how that compared in the moms and their fetuses that have been exposed, that have been exercises versus ones that weren't. And the exercised moms, the placentas had the diffusion distance took longer, time took longer, they had fewer blood vessels. It was interesting, the guy in charge of the research was invited to come give a talk, and they thought that exercise was good, and he went up there and showed the data where it was like, no, it decreased the amount of oxygen available to the guinea pig fetuses. And it's like, well, the lesson there is not to exercise or not exercise if you're pregnant, but to be sure that you don't start exercising. Okay, I'm 2 months pregnant, I want to be fit, I want to go start out running 5 miles a day. It's fine to continue with the exercise level you already have, because your heart is already used to doing that. It's not a new form of stress. The experiment was repeated with the guinea pig moms exposed to carbon dioxide. So hypoxia at 12%. Remember what normal is? 21% is oxygen. So they were put in tents of only 12% oxygen. And the opposite thing occurred. The fusion time decreased, and the placental tissue showed an increased number of fetal capillaries, and blood vessels. The research I worked with, we used carbon monoxide. There was no change. And I grew up with scientific method. You don't have preconceived ideas. You might have a hypothesis, but you don't have preconceived ideas about what you're supposed to get. So I got this no change, and so I'm writing my thesis and thesis and trying to figure out what the cause was. Remember carbon monoxide, you have normal PO2? So I was really excited. It's like, okay, well the PO2 must be, rather than the oxygen content, it must be the PO2 as one of the trigger factors on whether blood vessels are developed or not developed. because PO2 is normal, therefore no change. Whereas in hypoxic hypoxia, you would have a decrease in PO2. And it was much to my surprise, the overall primary investigator, the big honcho guy, was telling me I'd done the research on that this was not the right answer. And it really shocked me because I was like, well, I thought you were supposed to be open to whatever the data is and then try to figure out, you know, what happened behind that rather than, well, this is the wrong information, therefore the experiment was wrong. So it was very surprising to me as a 21-year-old to start arguing with this 65-year-old guru of fetal physiology. But so hypoxic hypoxia is either a drop in the value of oxygen, so you're in an enclosed space, you're not buried alive in a coffin, all right, or that we had the problem back in the, it made a movie out of one of the space shuttle, not space shuttles, but one of the spacecraft, I believe it was before the moon walk. There's a problem with the carbon dioxide converters.
Apollo 13.
Yeah, Apollo 13. They had to do some rigging to save their lives and get back to the dump in the ocean. And so that was an increase of CO2 displacing the oxygen as they used up the oxygen. Or it could be a normal amount of oxygen in the air, but a problem getting it across the respiratory membrane. Okay? So both COPD, both obstructive and restrictive diseases, could cause a problem with the diffusion. High altitude is what we're going to look at here because it's a more interesting kind of a problem. And any of you experienced acute mountain sickness or high altitude sickness? You know what altitude you were at?
It's probably about 9,000.
9,000?
It was when I was young.
Do you remember what the symptoms were?
Just huge headache and I got nausea.
Anybody else?
Yeah, in Cusco, when I was in Peru.
Yeah, in Peru.
Yes.
Okay.
We were at 4,000 meters of altitude.
4,000, so that's about a little over 12,000
Yes.
feet, because a meter is 39 inches instead of 36. So the values that see here, I was about 8,000 feet. I think I was in the wilderness area, up in the Sierra. And my husband, we were dating at the time, but decided to go camping for the weekend, and I hadn't backpacked in a long time. So he was carrying most of the stuff in his pack. We set up camp about 200 feet below the pass. I decided to climb up to the pass. And I was tired, but I didn't have a headache. And I took my pulse, and it was 210 beats per minute, which freaked Steve out a bit. And so he said, well, let's rest a bit and then go back down. And I got nauseated and diarrhea, and so he said, let's just go back to the car. And it was really weird, because just climbing 2 or 3 feet up to go over a boulder would make me nauseated. Not the altitude, but the exertion. So these numbers are not the air 66% oxygen, but remember the percentage is always going to be the same.
So of the 20% available.
21%. So 21%, 21%, 21%, 21%, but of fewer and fewer total oxygen molecules. So 100% here is going to be 160 millimeters of mercury of atmospheric PO2. Okay? This would be 84% of that 160. So if 100% oxygen is in your alveoli, in your blood, say a PO2 of 104, then at 5,000 feet you would have a PO2 of approximately 85, 86. And at 11,000 feet, a PO2 of maybe 68. So this is a value, and then you can see up at Mount Everest, at 29,000 feet, you have about a third amount of oxygen available to your tissues. So high altitude hypoxic hypoxia. This can lead to acute mountain sickness or high altitude sickness, and AMS stands for acute mountain sickness. All right, so a headache, a little bit of digestive problems are mild symptoms. Severe symptoms are classified as their effects on the brain and on the lungs. So it says here altitude cerebral edema. That's swelling, and it's not exactly known why we get cerebral edema. There's a retention of sodium, and the cells in the brain swell. We've got a skull, so it's not going to swell. And so that means that there is pressure being placed on the brain, so you get the headache, confusion, even loss of consciousness. With that increased fluid, we're diluting out the ions. So remember, we get action impulses potential because sodium ions at high concentration move into the cell. Well, if we're diluting that out, there's not going to be the same gradient, so there's a problem with action potential creation. Altitude pulmonary edema, high altitude, so this is HACE, H-A-C-E, and this is HAPE, H-A-P-E, high altitude pulmonary edema. So again, the causes aren't particularly known, but remember we talked about the perfusion, ventilation, coupling yesterday? And the fact that if one part of the lung has low oxygen content, the arterioles would constrict and divert blood flow to another part of the lung that has better ventilation. Well, if the entire lung has poor ventilation, these areas are not talking to each other. They each think they're, quote unquote, react as if they're the local poor ventilation and their blood vessels constrict. So when blood vessels constrict, what happens to hydrostatic pressure? It goes up as we decrease the radii, and so increased push of fluid out of the blood vessels into the alveoli, and you get pulmonary edema. Remember we talked about pulmonary edema with congestive heart failure? As pressure backs up in the left ventricle, the left atrium, and into the pulmonary vessels, it pushes out of the capillaries into the alveoli. That's going to increase our diffusion distance and make things worse as we have less oxygen diffusing into the blood. So those conditions are HACE, high altitude cerebral edema, and HAPE, high altitude pulmonary edema. So at about 8,000 feet, our PO2 is sufficient to, because of our lung plateau, to still saturate our hemoglobin above 90%. Once we get above 11,000, this is considered very high altitude. We have trouble saturation, especially with exercise, all right? And then above 19,000 feet, even without exercise, you're going to have difficulty saturating the blood with oxygen. So I have a couple of videos. Well, first, here's a story. This is Peru. This couple from dad and father, father and son from Canada, had a couple of years ago, had gone hiking in Peru, and they were hiking it's deeper than our Grand Canyon, but at an altitude of 5,000 to 11,000 feet. And they climbed down, and were climbing back up again. The dad was several yards ahead, and his group, he started having chest pain and collapsed. And they did 2 hours of CPR, but he died there on the trail. And they still had another 6,000 feet of elevation to carry him out. He had pulmonary edema, so it wasn't a heart attack. It was fluid in the lungs that he had developed at altitude with exertion. And about 1 person in 100 will develop high altitude sickness. And it has nothing to do with your physical fitness. I had 2 students, I had a student last spring, she and her boyfriend are planning on climbing Mount Everest, not this summer, but next summer. And they had been climbing Mount McKinley, that's Africa's, not Africa, Alaska. Or is McKinley in California? Whitney, McKinley's in Alaska. They climbed Mount Whitney, and she was doing fine. The ranger had told them to take like Excedrin, Tylenol ahead of time, that it would help them prevent mountain sickness. Dexamethasone is another drug that you can take. And she'd done it, her boyfriend hadn't, and they were not quite at the top. And he started hallucinating. He saw a log cabin that he wanted to go to. The only problem was off the trail in the middle of the valley up in the air. And he wanted to hike off the trail and head over to that log cabin. And he was more physically fit than she was, but was affected by the altitude. And with pulmonary edema, with cerebral edema, you lose your ability to maintain logical thought processes. So here's a picture of a guy.
Need